Welcome everyone to the Science of Childhood Cancer lecture series hosted by the St. Jude Comprehensive Cancer Center here in lovely Memphis, Tennessee. I'm Paul Northcott, and I'm an associate member in the Department of Developmental Neurobiology and a member of the Brain Tumor Program here at St. Jude. It's my pleasure today to welcome one of my longtime friends and colleagues, Dr. David Jones from the German Cancer Research Center in Heidelberg. And so without any further delay, it's really a pleasure to welcome David to this exciting virtual series. David is currently a group leader in pediatric glioma research at the German Cancer Research Center in Heidelberg. He earned his PhD at the University of Cambridge, where he trained under Peter Collins. And during his PhD, he made a seminal finding that tandem duplications produce a novel oncogenic BRAF fusion that define the majority of pilocytic astrocytomas. And this is one of the most common childhood brain tumors. In 2010, David joined the DKFZ as a postdoc in Stefan Pfister's group, where he was assigned as a lead biologist to the International Cancer Genome Consortium, or ICGC, PedBrain Tumor Project. And during that era, it was a really exciting time. The genomics era was really exploding, and applications of whole genome sequencing, RNA sequencing were really enabling new discoveries across all cancers, let alone in childhood brain tumors. And David played a pivotal role in several high-impact publications during that era, including the discovery and description of chromothripsis in Li-Fraumeni syndrome sonic hedgehog medulloblastoma. And he also published the first phase whole genome sequencing studies of medulloblastoma and pilocytic astrocytoma. In 2012, he became a subgroup leader again at the DKFZ, where he began to lead a group primarily focused on low-grade and high-grade gliomas. During this era, David played a major role in several genomic-related studies, and one of the most impactful was the application of DNA methylation-based classification of brain tumors. And I think you'll see some of that work presented here today. This has now become a gold standard molecular-based test in neuro-oncology and has rapidly changed how brain tumors are diagnosed as a companion to conventional neuropathology. In 2019, David became an independent group leader at the DKFZ and HOP Children's Cancer Center, where he now leads a very productive group focused on glioma, molecular diagnostics, and precision medicine. He's the recipient of numerous awards that I will just name a few: the DKFZ Alumni Award, the Kind Philip Prize for Research in Pediatric Oncology, and he's authored more than 250 publications at this young stage of his career, making him one of the most heavily cited scientists in Germany, and he's not even 40 yet. As I said, I've known David for many years, more than 10 years now. It's really been a privilege to be a colleague and friend of his. He's an outstanding scientist, and I'm really excited to hear some of his latest work here today. And so without any further delay, we'll let David get started.
Thanks very much, Paul, for the extremely kind introduction. Yeah, not quite 40 yet, but it's creeping up on me, unfortunately. So I'll be catching up with you soon. Yeah, so thank you very much again for the introduction, and thanks for the very kind invitation to speak in this series. It's really my great pleasure and honor to be here this evening, my time, early afternoon over there, to join you for this seminar series. And I'd like to present some sort of background briefly on some things that have been published recently, and then also towards the end show some unpublished data as well around classification of pediatric high-grade glioma and some novel, interesting new subgroups within pediatric high-grade glioma. So I know that the audience for this series is not necessarily brain tumor specific, so we'll start with very brief sort of basic introduction on pediatric brain tumors and pediatric gliomas. So as a reminder, brain tumors relatively compared with adults are much more common in children, so the second most common tumor type in children, with a slightly higher incidence in boys than in girls. And despite the slightly lower incidence than leukemias, they're the leading cause of cancer deaths in children. So both in younger childhood ages, but also in teenage and into young adults, in males and females, a leading cause of cancer death. So really one of the major unmet needs still in the field of pediatric oncology. I think what can be seen here is that we're really facing an issue now where survival rates for brain tumors are stagnating. So if you look at leukemia, for example, 60, 70 years ago, 5 year survival was way down at 10%, but has really improved dramatically and is still improving, is now around about 85%, or even 20 years ago was already 85% 5 year survival. Whereas brain tumors started off a little bit higher up this curve, around the 35% mark, and did improve gradually up until around about the turn of the millennium. But since then, we've pretty much been stuck at this 70% 5 year survival level, and that's really not changing anymore. So we've reached this plateau, unfortunately, where there have been no major changes in increasing survival over the past few years. And one of the major causes of that are the pediatric gliomas, which make up about half of all pediatric brain tumors and split into multiple different entities and subgroups. So the biggest single entity being the pilocytic astrocytoma, low-grade tumor, but multiple other groups of various malignancy grades within the pediatric glioma family. And they come in two main flavors, essentially. So low-grade glioma being the most common childhood brain tumor with a very good overall survival, but this means that the tumors often become a long-term chronic disease where the patient is suffering from long-term effects, not just of the tumor, but also of the treatment for that tumor. And here, improving quality of survival is the most important factor now. So we've moved away from trying to improve the quantity of survival rates for low-grade glioma already being over 95%, but trying to improve the quality of life for survivors is now a major focus. But I'm not going to speak really much at all about low-grade glioma today. And the main focus is going to be on the high-grade gliomas, which are really uniformly lethal, so very, very poor outcome, extremely heterogeneous, and that's going to be the main focus of this presentation today, this extreme heterogeneity, which makes omics profile, it makes tumor modeling, and it makes planning of clinical trials extremely challenging. And I think that's something that we really need to address now to be able to more systematically and in a structured way start to make progress. So the high-grade gliomas are something like 15 to 20% of all pediatric brain tumors if you take typical glioblastoma as well as diffuse midline gliomas and the malignant glioma NOS groups together. And as I mentioned, prognosis of these tumors is really extremely poor. So average survivals of around about 12 to 18 months, depending on the precise molecular subgroup that we're talking about. And it's been known really for many, many years now that pediatric high-grade gliomas clearly show differences in their biology compared with adult high-grade gliomas in multiple different levels. So copy number is shown here, expression with these two studies now going back 10 and even 20 years. It's been known that they've shown differences with the adult disease. And that was really shown really without question and finally categorically in 2012 with these two parallel identification of recurrent histone mutations in histone 3 in pediatric high-grade gliomas. So these two studies, one led by Nada Jabata in Montreal together with the group of Stefan Pfister, and also the very nice study from St. Jude's from the Pediatric Cancer Genome Project, identifying these two hotspot mutations in the tail of histone 3, which are now together known as the oncohistones, which are driving a substantial part of pediatric high-grade glioma. And I think really at that point became, if it wasn't already, became really abundantly clear that we have to start thinking about pediatric high-grade gliomas as really a separate disease from adult glioblastoma. So at the same time as those, or shortly after those mutations were identified, it was also demonstrated that those tumors not only differ in the mutational status, but also have fundamentally different epigenetic signatures. So you can see in this DNA methylation heat map here the IDH mutant tumors, including a few of the older teenage patients, but really the K27M mutated and the G34 mutant tumors, primarily in pediatric, very different in terms of their epigenetic profile from the more common adult glioblastoma. Unfortunately, however, despite these multiple bits of evidence that have been collected over the past years, it's taken quite a lot of time for this to filter into the classification systems that are used for brain tumor diagnosis and classification. So in the current version of the WHO CNS tumor classification from 2016, of the different pediatric subtypes, only the H3K27M mutant tumor is considered as a separate entity at the moment. And the rest of pediatric high-grade glioma is just mixed in with the adult disease with a very brief discussion on the differences between pediatric and adult high-grade glioma. But despite the fact that it's still not yet gained formal recognition, there has been a very major focus in recent years to continue to explore this heterogeneity in pediatric high-grade glioma and try to really refine the definitions of molecular subgroups of this disease. One of the earliest studies to look at that from Andrei Korshunov in Heidelberg here for a few years ago now showed that in addition to the typical high-grade groups, so the histone mutant and histone wild-type true high-grade gliomas, if you just take an unselected series of histologically diagnosed high-grade glioma in children, what we see is that there's a proportion of those tumors which have molecular features common to actually more lower grade tumors. So ones which really look true low grade, including with the presence of the BRAF fusion, for example, or tumors which resemble molecularly more PXA-like signature, which almost all harbor this co-alteration of the BRAF V600E point mutation with homozygous deletion of CDKN2AB, which again are hallmark features of PXA. So that it's important to recognize that not everything which is being histologically diagnosed as a high-grade glioma will necessarily behave clinically as a high-grade glioma. And there might be ways to pick out some of those tumors based on the molecular profile, which may have a better outcome down the line. That was then followed up with this very large series put together by the group of Chris Jones in London a few years later, where they brought together data from multiple different publications and ended up with more than 1,000 cases of high-grade glioma with molecular profiling and also clinical annotation data. And here they identified 5 different groups within the histone wild-type high-grade glioma subset. So including the PXA and the low-grade groups, which also in this analysis were confirmed as having a superior outcome from typical pediatric high-grade glioma. One group, which was also found to be specific to infants, so very, very young patients, this small group here. But still at this stage, the rest of the histone wild-type tumors were really thought of as being a relatively sort of lumped together with this umbrella-like term of histone wild-type, if you like. Just to segue briefly onto that infant-specific high-grade glioma group, this is now published, so I won't dwell in great detail, but a study both from the Toronto group led by Uri Tabori and Cynthia Hawkins, as well as a collaboration between London, Heidelberg, and St. Jude's, showed that this group of infant hemispheric glioma in very, very young patients, so really the peak in very young age, typically below 2 years old, have a clearly different molecular profile. They have a superior outcome. So here the IHG for infant-type hemispheric glioma with clearly better outcome than typical high-grade glioma. But the most interesting from the biological and also from the treatment perspective is that many of these tumors in these very, very young patients have fusions in one of these genes here. So one of the NTRK family, NTRK 1, 2, or 3, in ALK, in ROS1, or in MET. So a high proportion of this infant group showing these potentially druggable fusion events, which are now being investigated or will be soon in clinical trials with targeted inhibitors against those fusions in that patient population. So to come back to this overview, we have these histone wild-type groups here, the PXA-like, the low-grade, that infant group, and this histone wild-type group. But really, this was still just the tip of the iceberg in terms of the amount of heterogeneity that we see in pediatric high-grade glioma. I want to explore that in a bit more detail now on the following slides. So you won't be surprised that I'm going to mention briefly DNA methylation analysis of brain tumors. And as Paul already mentioned, this is now really changing in many ways the way in which brain tumors are being investigated, both for research purposes, but also as a diagnostic tool. And we think that the way this works is that these methylation marks on the DNA are marking the epigenetic state of the cell of origin of a given tumor. So if you think about the different stages that a cell will go through as it starts from an early stem cell becoming more and more specialized and differentiated, at each step along that lineage, all the cells are also changing their epigenetic makeup and the way that the genes are switched on and off to be able to change their specialization in that way. And at each stage, you have then a very, very distinct epigenetic state, which marks a particular cell type. And a tumor arising from a given precursor cell type essentially then becomes a clonal expansion of that epigenetic state. So instead of seeing in a sort of normal brain tissue analysis, a mixture of epigenetic profiles from very, very many different cell types, what we think you're primarily seeing in the tumor epigenome is this purified expansion of the epigenetic profile of one precursor state, which is why by measuring the DNA methylation, we think you can very clearly distinguish between different tumor types based on that memory of the cell of origin. And this was shown at the beginning of 2018 in this study looking at more than 80 different brain tumor types with DNA methylation array analysis, showing that that seems to, even on this large scale, really hold true, that each different tumor subtype has its distinct DNA methylation profile here, that different tumors from different patients of one subtype cluster together with each other in terms of their epigenetic profile, but distinct subtypes of the tumors are can clearly be distinguished from each other. And it's this principle that is now being applied also to assist in the diagnostic process. And at that time, when we were assessing this series, we saw that by adding this tool on top of a standard neuropathological workup, we would get a change in diagnosis in around about 10 to 12% of cases. So we really think that this tool can assist with defining molecular patterns specific for different tumor types, which help in the diagnostic process. And we're now continuously expanding the database of DNA methylation profiles that we have available to us in Heidelberg. Currently, that stands at more than 85,000 DNA methylation array profiles that we have in that database, including about 60,000 brain tumors. And this is really, as you can imagine, an amazing resource, and I'm extremely grateful to be able to work with this data on a daily basis, to be able to not only explore substructure within some of the known tumor types, but also for identification of novel brain tumor subtypes, which are so rare that it's simply not possible to define those purely based on the histology or by noticing similarities in microscopic appearance. You really need to get these very, very big numbers together to be able to start having this power for novel discovery. So when we look in detail at this very selected set of high-grade gliomas now, we see that far from being just 5 histone wild-type subgroups, there's actually a huge amount of heterogeneity within what is histologically being called high-grade glioma in children. So very, very many different subgroups. I won't go into all of them today, but I will pick up on a few which I think have either biological and or clinical relevance to be aware of. So starting with a diffuse midline glioma H3K27 altered, this is the most lethal form of pediatric high-grade glioma, with tumors almost exclusively located in the midline and especially in the pons, and a very high frequency of the K27M variant of the histone 3 mutation, which I mentioned earlier on. And there's a lot more detail known about the mechanism now, which I won't go into here, but some beautiful work that's been published from multiple groups in recent years. But essentially, you can think of the K27M mutation in terms of the structure of the peptide as mimicking a monomethyl lysine at that K27 position, which inhibits the activity of PRC2. And one of the clearest consequences of this mutation is that the tumor cells completely lose trimethylation at the histone 3 K27 position. So just one example here of specific antibody staining for the mutation itself. But when you stain for the trimethylation of histone 3 K27, you see that in the tumor cells, you have this pale blue negative staining here, where really almost completely there's a loss of this mark. But what's now been seen is that there's also a subset of these diffuse midline gliomas which show the same pattern of complete loss of K27 trimethylation, but without showing the K27M mutation. And in this publication led by a group of Jacques Grill in Paris, it showed that those histone 3 wild type diffuse midline gliomas, which don't differ in terms of their survival from the tumors which have the K27M mutation, show a substantially elevated expression of a gene which is now known as EZIP for EZH interacting or EZH inhibitory protein, previously known as CXOR67. And that was actually first described in the posterior fossa A subgroup of ependymoma. And it was shown that the structure of CXOR67 or EZIP here on the right mimics the structure of the K27M mutation in the histone tail. So this is H3 with the lysine 27 methionine mutation. And this is the structure of the naturally occurring sequence of EZIP. And you can see very, very similar structure, including this methionine here. And it's thought that overexpression of this gene is essentially another way of blocking PRC2 activity, which is why also in the tumors with overexpression of this gene, you get this pattern of complete loss of the K27 trimethylation. That mechanism of overexpression of EZIP, but also the K27M mutation itself, have now both been found also in a distinct subset of thalamic gliomas, so outside of the pons, but still in the midline. And interestingly, these were seen to very, very frequently harbor a co-mutation of EGFR. So this group was first described by the UCSF group in the middle of last year, with biothalamic gliomas and frequent mutation of EGFR, which was then followed up on by Philip Sievers in the neuropathology department here in Heidelberg, and shown that this is really a clearly distinct subset of midline glioma, where you get some tumors with the K27M mutation, and the tumors of this group which don't harbor the K27M mutation have overexpression of EZIP to the same level as those H3 wild-type diffuse midline gliomas, which I just showed. And again, very, very frequent alteration of EGFR, which is extremely rare in the other K27 altered diffuse midline gliomas. So this co-alteration of K27 plus EGFR is highly specific for this relatively small thalamic subset. So now what we see is diffuse midline glioma is not just the K27M mutation, but we have diffuse midline glioma K27M, including the classic diffuse intrinsic pontine gliomas. But a subset of tumors, particularly in the pons, have, instead of the K27M mutation, overexpression of EZIP, and very often mutation of ACVR1 as well. And then we have this even more distinct subset up here, DMG EGFR, which have either the K27M mutation or EZIP, and together with EGFR mutation. And in terms of location, in the thalamus, still majority of the tumors are histone 3.3 mutated, but the significant minority of tumors in the thalamus belong to this group with EGFR mutation. And in the pons, again, still mostly H33 K27M mutation. So in terms of absolute numbers, that's still by far the most common, is that you have the typical histone 3.3 K27M mutation, but also in the pons, several with histone 3.1 instead of 3.3, which tend to have this ACVR1 mutation. And occasionally in the pons, rather than the histone mutation itself, the overexpression of EZIP, and again, those also very often have the ACVR1 mutations. So switching back to histone wild type, also here there's a lot of substructure that can be seen. And the three most common groups in terms of the sort of broad structure have been referred to in different ways. For these purposes, I'm going to use the names which came from the study of Andrei Korshunov: pediatric RTK1, pediatric RK2, and the MYC-N group. You can see very distinct from each other. The pediatric RTK1 is frequently associated with amplifications of PDGFR alpha. Pediatric RTK2, often with amplification of EGFR, and the MYC-N, as you would expect from the name, with amplification of MYC-N, and actually very frequent co-amplification of both MYC-N and a gene which is located very nearby called ID2. So displayed again here, so in the MYC-N group, around about half of those tumors have a MYC-N amplification, very rare in the other two groups. In the RTK1 subset, about a third have amplification, but very rare in the other two groups. And in the RTK2 group, again, about half with EGFR amplification, and also of interest, very frequent homozygous deletion of CDKN2AB at 9p21, which is also much rarer in the other two subtypes of pediatric high-grade glioma. In contrast to what's seen in adult glioblastoma, and also in contrast to the thalamic EGFR mutated group, which has a very poor outcome, there's actually some hints that the RTK2 group in the histone wild type setting, so this pediatric RTK2 group, might actually show a slightly better outcome than the other two histone wild type groups, with the worst prognosis being seen in the MYC-N amplified group. And this is essentially equivalent to the K27M mutant DIPG. So the MYC-N amplified really being the extremely poor actors with the dismal survival here. The pediatric RTK1 subgroup is the most common of the histone wild type groups. And I think it's also, in my eyes at least, the closest to what might be considered a typical histone wild type pediatric glioblastoma. So in terms of trying to define a group to act as a wild type control, for comparing with the histone mutant tumors, I think this RTK1 or PDGFR alpha group would be the one which is probably what has the majority of the features which would be considered to be a typical pediatric glioblastoma. So a standard copy number profile, as you can see here, again with this amplification of PDGFR alpha. And what's interesting is that other phenotypic aspects of pediatric high-grade glioma have now also been associated with some of these different subsets of the histone and IDH wild-type group. And that includes the germline mismatch repair-associated high-grade glioma, which display a hypermutator phenotype because of the hereditary defects in DNA damage repair. And this study from Andrew Dodson and the group of Ori Tabori again in Toronto. And what you can see here is that wild type C, according to the names of the meta-analysis from the London group, are in the light blue dots here, and the dark green are the tumors with hereditary DNA damage defect, and those essentially cluster together. So most of these mismatch repair associated hypermutated tumors match with this pediatric RTK1 subgroup. And that's also true of secondary glioblastoma as a result of radiotherapy for medulloblastoma or leukemia. So these radiation-induced secondary glioblastomas also seem to belong primarily to the pediatric RTK1 group. Again, shown here in black, the radiation-induced gliomas after a prior medulloblastoma. The empty circles here are the radiation-induced gliomas after prior treatment for ALL, and in green, again, the RTK1 subgroup. And you can see that they don't form distinct subsets from each other, but rather these radiation-induced tumors are mixing with the non-radiation-induced RTK1, so the sporadic RTK1 tumors, and also show very, very high frequency of PDGFR alpha amplification, which fits with belonging to this subset. Slightly higher frequency of MET amplification than is seen in non-radiation related pediatric tumors, and very high proportion of CDKN2AB deletion, which is actually not so common in the pediatric RTK1 group overall, but is very frequent in these radiation-related tumors. And very, very similar findings have been seen in a parallel study led from Denver and from St. Jude's, which also showed that the treatment-induced high-grade glioma in black here show very, very similar DNA methylation profile with the pediatric RTK1 group. Diving a little bit deeper yet still, there's even now some evidence for substructure within the RTK1 group. So it's a little bit reminiscent of the medulloblastoma story now, where it started off with the 4 consensus subgroups. And as we go deeper and deeper and collect increasing numbers of molecular profiles of those tumors, we can see that those major subgroups can be split into further subtypes, which also have clinical and molecular relevance for being further subdivided. So we're now starting to think of the RTK1 group as potentially being split into the major group, which is RTK1A, but also some additional subsets of RTK1B and 1C. And the mismatch repair tumors and the radiation-induced tumors are not randomly distributed amongst those different subtypes within RTK1. So you can see that essentially all of the mismatch repair-associated tumors are falling in this bulk RTK1A subtype here, whereas the radiation-induced tumors still belong to this RTK1 family overall, but they're now distributed within the 1B and C subgroups, and only one example of a radiation-induced tumor in RTK1A. So clear link between these phenotypic or causal subsets of pediatric high-grade glioma with RTK1, but not randomly distributed throughout that EPA layer of substructure. And again, I've shown this already, but the histone wild-type tumors are more complex still. And I wanted to pick up on a couple of groups just briefly now that are starting to come out of these analyses of identification of novel subsets. And one here, a group of just over 60 cases that we've collected, distributed throughout the brain anatomically, various different histological diagnoses, but the biggest proportion being histologically diagnosed as high-grade glioma. But when we looked in the RNA data of this tumor, what we saw is that all of the cases where we're able to get RNA data showed a fusion either with of PATS1, either with MN1, shown here, or with EWSR1, so the partner of the typical Ewing sarcoma FLE1 fusion, but here with PATS1 as the partner. And both fusions retain the zinc finger binding domain of PATS1 with the upstream activation domain of EWSR1 or MN1. So probably PATS1 is the primary partner driving the biology of this group, and you need the PATS1 DNA binding domain with some kind of transcriptional activation domain to lead to the downstream effects. What's not clear, though, is whether we should really be considering this a glioma, despite the fact that most of them were histologically diagnosed as glioma. On the expression level, we see very low expression of GFAP, very low expression of OLIG2, notably quite a high expression of CSPG4, which is also known as NG2, which might be not entirely specific, but a useful marker for picking these up in terms of immunohistochemistry. And also some other markers which are a bit more specific. So IGF2, very high. The PAX2 transcription factor, very high, and also the PAX1 gene itself, clearly more highly expressed in this group compared with a variety of other pediatric glioma groups. So maybe neuroepithelial might be a better term than glial here, but an intriguing group. There's a difference in the age of onset between the MN1 PATS1 and the EWSR1 PATS1, so the MN1 being in slightly younger patients, but no obvious difference in outcome between those two subsets, but a clear difference in outcome from typical high-grade glioma. And again, remember, many of these were histologically diagnosed as glioblastoma, but in terms of overall survival, small numbers, but a hint at least that they're performing better than a typical high-grade glioma. The second subset is this group that's labeled PLAG here, which is a subset of about 30 cases. And those have the unifying feature of having an amplification either on chromosome 6 here, around the PLAG-L1 locus, chromosome 20, where PLAG-L2 is located, or very rarely on chromosome 8, where PLAG1 is located. So this PLAG family of transcription regulators. PLAG-L1 is broadly expressed in many normal tissues, including the brain, and is linked with neural precursor development and differentiation in the brain. Whereas PLAG-L2 and PLAG1 are usually only expressed in fetal tissues. And if overexpression there has been shown to block differentiation. So slightly different roles depending on the context, that they seem to show similarities in terms of their broad molecular profiles. PLAG-L1 group may be slightly older patients, PLAG-L2 may be slightly younger patients, although the numbers are still a little bit too small to make any definite conclusions on that as yet. In terms of expression, again, very, very high levels of IGF2, intriguingly, and also high expression in this subset of the RET oncogene. And when we looked in a bit more detail at that, what we could see is that in these PLAG amplified tumors, compared with other pediatric RTK tumors, based on enhancer marks here, there's a very strong signal for K27 acetylation in the PLAG-amplified tumors just upstream of the RET gene, which is not seen in the other pediatric high-grade glioma, and in the PLAG-L1 amplified tumors, binding of PLAG-L1, and in the PLAG-L2 amplified tumors, binding of PLAG-L2 also at this enhancer locus. So there seems to be direct association of this amplification of PLAG-L1 and 2 with this enhancer mark driving the expression of RET in these tumors, potentially as a novel therapeutic target in that subset. So where are we now in terms of pediatric high-grade glioma classification 2021? So there will be a new edition of the WHO classification very, very soon. There is now finally a distinct chapter for pediatric type diffuse high-grade glioma, which is certainly progress in itself, and really an acknowledgement of this clear distinction between adult and pediatric high-grade glioma, but overall still a little bit less progress in terms of recognizing high-grade glioma subtypes than for pediatric low-grade gliomas, for example. And we're at the stage now where the K27 altered, the G34, and the infant type groups are acknowledged as separate entities, but unfortunately still only one real category for all of the other very heterogeneous H3 and IDH wild type tumors. There's still a bit more work to do there. So thinking about where that might be going in the future, if we take all pediatric type high-grade glioma, or including potentially some high-grade neuroepithelial tumors, because they're often histologically diagnosed within this high-grade glioma group. Again, the one important point to note is that a subset of what's histologically called a high-grade glioma will be molecularly a lower grade glioma, either similar to pleomorphic xanthoastrocytoma or to real low-grade tumors like pilocytic astrocytoma. But within the higher grade lesions, we've got now pediatric type diffuse glioma H3 altered, including the tumors, the diffuse midline tumors with K27 trimethylation loss, and identification of subgroups within that subset, and what is still at the moment considered diffuse glioma, H3 G34 mutated, but some very nice work from Nada Jabado and Claudia Kleinman has shown now that actually these come from an interneuron progenitor, have almost no OLIG2 expression. So whether they should really be considered glioma is questionable at this stage. So the G34s might sort of skip off into a parallel branch here. In terms of glioma IDH wild type, we've got the major groups of the RTK1, RTK2, and MYC-N, really seeing some substructure also within those now. Went into a little bit of detail about the RTK1A, 1B, and 1C, and their association with the hypermutated tumors and irradiation-induced tumors. We also see a similar substructure now within RTK2, between RTK2A and B, and a clear recognition that the infant type tumor is a distinct entity, frequently associated with some of these, fortunately, potentially quite druggable fusions as a recurrent event in that subset. And then for those tumors being diagnosed as glioma, but probably again representing more a neuroepithelial tumor, more primitive neuroepithelial rather than a glial origin, we have the neuroepithelial tumor PAX1 fused, neuroepithelial tumor PLAG-L1, L2 amplified, and I think we'll see several similar groups still to come over the next few years that will be identified as the profiling gets better and as the case number increases. And I think that really now the next generation of analyses of pediatric high-grade glioma are already well underway in terms of single-cell analysis, in terms of preclinical modeling and drug testing, including looking at the interactions between the microenvironment, and tumor cells. But I think that all of these efforts need to be assessed in a subgroup specific manner. So we need to take account of this tremendous heterogeneity and the fact that there is substructure within what's being called pediatric high-grade glioma to help to better interpret some of the data coming out from those really exceptional efforts that are underway in several sites around the world. So with that, I'd like to sum up and really highlight that pediatric high-grade gliomas are extremely diverse and, at least in my opinion, biologically very intriguing. I think there's a lot of features of high-grade glioma which go far beyond pediatric tumors in terms of their impact on the field of oncology, chromatin biology, and other areas. Many more subgroups have now been identified than was previously anticipated, and each of those subgroups can be linked with distinct driver mechanisms, even for subtypes within known subgroups, such as the K27 trimethylation loss group. Important, very important clinically to recognize that not all histologic high-grade gliomas are necessarily true high-grade gliomas. Some with more low-grade molecular patterns, and some probably not gliomas at all. And I think also it's really not a case of subgrouping for the case of subgrouping. I don't have some kind of weird fetish about splitting tumor entities into smaller and smaller groups just for fun, but I think knowing in more detail about what we're facing here allows for better identification of therapeutic vulnerabilities, better modeling of the disease, and also better planning of clinical trials. So some of these groups might be associated with therapeutic targets, which might be 100% of one particular subset, but so rare overall that they would have been missed when treating all high-grade gliomas together. But unfortunately, as with many of the more malignant pediatric entities at the moment, the knowledge about the biology is rapidly outpacing the rate at which we can convert it into clinical benefit. And I think that's where the focus needs to shift now, is in really getting the hands back in the lab and converting the knowledge into functional screening and hopefully into new treatments in the future. So with that, I'll stop. Thanks to my group, particular thanks also to other groups in Heidelberg, very, very close collaborators, and other groups around the world who've contributed with samples, with comments, with collaborative studies. And thank you for your attention, and happy to take some questions.
Great, thank you, David. We have many questions in the chat, and you've done a great job of leaving time for those questions. So we will start reading through those now and ping them your way. So starting at the top, in no particular order, do you also observe G34W variants of H3.3?
Not in the brain tumors, interestingly. So they're common in some subtypes of bone tumors. But as far as I know, we've not observed a G34W in brain tumors. So the R is the most common. G34V is also seen occasionally, but W seems to be bone specific for whatever reason.
Great. Okay, next question. Very nice talk. Can you tell us about the differences between pediatric glioblastoma RTK2A and RTK2B?
Yeah, I would love to be able to do that indeed. Question from David Ellison, so putting me on the spot. At the moment, we don't know. It's clear that EGFR plays a very major role in that group, even more so than we had previously identified. So in the beginning, we had primarily just the copy number data, and we could see that EGFR amplifications are enriched in that group. What we see now is that many of the tumors which don't have an EGFR amplification have an EGFR mutation instead. There's potentially some hints that those mutations are not the same types of mutations as we're seeing in the thalamic K27 trimethylation loss group. So there it seems to be the A289 hotspot or an hotspot in exon 20. In the RTK2 group, it seems to be a bit different, but what's really different between A and B, we are not there yet, but we're hopefully working on it.
Great. So a very closely related question. Do you have experience in EGFR inhibitors for DIPG with EGFR exon 20 insertions?
We don't have any experience of that clinically. I think it's very, very early at this stage. It's a relatively recent finding. In that initial paper, which I mentioned from the UCSF group, they had a couple of patients in that series that had received EGFR and targeted inhibition as part of their therapy, and some anecdotes of some responses also to those inhibitors. I think at the moment the numbers are very, very small, and but that's something that would be, that's one subset where I think it clearly makes sense to start planning a trial.
Right. So a technical question here for you. Based on the massive methylation cohort that you've put together, do you think some intra-subgroup clustering could be driven purely by CNVs? And could regressing out CNVs facilitate novel subtype identification?
That is a very technical question. To my knowledge, and please don't hold me to this, I don't think we've seen, at least we haven't seen clear evidence for substructure which is purely CNV driven. And I think part of the reason for that might be that methylation tends to be really binary. So at most CPG sites in the genome, you either have zero methylation or a full methylation. There are not many sites which have one allele methylated and one allele unmethylated. So I think that when you do get copy number changes, you tend to retain that binary status of the mark that you're looking at. So either both copies were unmethylated originally and now all 5 copies are still unmethylated, or both copies originally were methylated and now you only have 1 copy but it's still fully methylated. So I think that in comparison to things like expression, this on-off nature of methylation makes it a little bit more robust to copy number, but I wouldn't exclude, as we learn more, that we might start to see a signal there as well.
Right. Okay, so a question from Dr. Susie Baker. It's intriguing to see the range of distinct subgroups emerging in the context of tens of thousands of methylome profiles in your data set. How much clinical data is available to connect with these data to allow you to identify subgroups associated with particular targets or responses? What is the capacity in Europe to use this type of data to stratify extremely rare tumors into structured clinical trials?
Yeah, very good question, and multifactorial question. So the clinical data is certainly much, much poorer than the molecular data. So I'll be very open about that, that for many of the tumors, we only have the methylation profile, which also gives us the copy number profile, but very, very limited clinical annotation. I think in terms of target identification, it's not so bad because some of the targets you can imply from the copy number status. And for many of the tumors, we do now have at least a gene panel sequencing on top of the DNA methylation. So identification of structural changes or point mutations acting as therapeutic targets, I think we would see. The hardest part at the moment still is linking that with outcome and having the numbers from the clinical data side to say what the prognostic impact of the subgroups is. In terms of clinical trials, it's becoming a bit more standardized now in Europe, I think. So we're seeing more and more groups starting to include not just the methylation analysis, but also broader sequencing analyses of DNA and also of RNA, importantly. So many of the good drug targets seem to be RNA-based, some of the kinase fusions. And I think as that profiling is becoming more widely available and not just in a research setting, but in a slightly more routine clinical setting, we're able to identify these groups more now also prospectively and not just in a retrospective way. So I think we're not there yet, and some of these groups are extremely rare to the point where you would never be able to plan a trial just for that group because it's 10 cases from 80,000. It would take 50 years to design a trial. But I think some of the groups where we're seeing 50, 60, 70 cases now in that larger database, now that we know what we're looking for, we're also seeing maybe every 2 or 3 months a new case coming in, either through us locally or through one of the collaborators, where we see, that also belongs to that group. And I think those are the groups. So things like the PAX1 fused group is actually not that rare. So we're probably becoming aware of one every couple of months. It's just that people couldn't pick them up before because they're so heterogeneous in terms of morphology. And if you don't know to look for that fusion, you wouldn't see it.
Right.
I think some of those groups you might be able to start planning a trial.
Yeah, some of those new fusions are really exciting.
Yeah.
And amazing that you can discover them amongst the 80,000 other profiles that are populating your atlas. So time for one or two more questions here. In view of the increasing granularity of closely related subtypes that presumably have the same cell of origin, how much of the methylation variability is due to cell of origin versus downstream effects of oncogenic drivers? That's the first part of Charles Mulligan's question.
Yeah, very nice question. So we think that on this sort of really zoomed out view, which I show when you just look at all 80,000, most of what we're seeing there, I think, is cell of origin. There are some notable examples to that. So IDH is an example where we know the function of the IDH mutation has a big effect on the epigenetic machinery downstream, and you get very, very large scale global methylation changes because of the somatic driver in that tumor. So there we see a mixture of somatic changes to the epigenome and the signature of cell origin. But for most groups, I think it's primarily cell of origin. But we're starting to see that a bit more now when you really zoom in. And I think that's the question when you go into this very granular level. Can you see signal on top of? So these 5 subtypes might share a cell of origin, but is it other things which are driving the subtypes within that subgroup? And I think that we are seeing some evidence of that now, that you see enrichment for maybe V600E in one set and FGFR in another set, for example. So similar pathways, similar origin, but slight differences in how those group based on the driver as well.
And just a related question to cell of origin versus methylation, et cetera. Does differential methylation correlate with deregulation of gene expression in your experience?
Yes, to a smaller extent than we would have expected, I think. So for some genes, you do get a very nice correlation that if you plot correlation between methylation and expression, you clearly see that they're linked. I think that's not the case for every gene. There are some features controlling expression which are much more relevant than the methylation. And I think that it's also the dynamic range that you're looking at in those two things is also very different. Methylation, you have, again, between 0 and 1, and if you do whole genome bisulfite sequencing with 10,000-fold coverage, for example, you could get very small increments on that 0 to 1 scale, but it's still in a small-ish range. Whereas expression, when you go from no expression to extremely high expression, that's over a much, much broader range than the methylation. So there is correlation, but not as much as we would expect.
As expected.
On a different
Right.
level. Yeah.
Great. All right, so we're just past the top of the hour now. So I'd like to officially close the event and once again thank Dr. Jones for his exciting talk and sharing some of his latest unpublished findings, and certainly look forward to reading about some of those new subgroups and subtypes and the fusions that drive them in the near future. He has kindly agreed to remain on this session to answer additional questions. So those interested in continuing to ping David with questions, please feel free to stay on for a few more minutes. And otherwise, we thank everyone else for joining and hope that you will join this seminar series for future events. We are reconvening next week when we have Karen Vuston from the Francis Crick Institute speaking on the role of metabolism in cancer. And so we thank you for attending this session today. Otherwise, we'll just keep going with questions now, David. Okay, so I've got time to ask a few more here. Could you elaborate on potential mechanisms of CDKN2A homozygous deletion in radiation-induced gliomas? Does radiotherapy actually induce this alteration?
Good question. The short answer is we don't know, but I will wildly speculate that we see generally slightly more structural rearrangements in the DNA in the radiation-induced tumors compared with sporadic tumors. And I could certainly imagine that the radiation is directly inducing some double-strand breaks. And at some point, you're not repairing those efficiently. And that some of the structural variations might really be as a result of the DNA damage caused by the radiation. But we don't have any functional experiments to prove that's the case. So that would really be still a speculation at the moment, or a hypothesis, but not proven.
Great. So I have another question here. Whether or not you know the answer to, I will find out. So has it been confirmed experimentally that EZ-HIP overexpression contributes to either tumor maintenance or methylation subgrouping?
Ooh, it may have been in a ependymoma. It has not been proven experimentally in high-grade glioma yet, but there are some cell lines and PDX models floating around which have now been identified as belonging to that group with EZ-HIP overexpression. So
it can be tested.
But also others are now starting to look at that, I think, and that's something that will be in the next round of experiments, definitely.
Okay,
I'm up to speed with the ependymoma literature there. It could be that they've done it already, but
it's evolving very fast. Okay, so another question from earlier. May I ask about treatment-induced epigenetic methylation changes in tumors and your thoughts about the use of such information? How stable are these signatures?
They're extremely stable, actually, surprisingly stable. So most of the tumors that we see in the brain tumor context are not getting azacitidine or other things that you might expect to have a direct effect on DNA methylation. So what we see typically going from primary to relapse, even if that's 5-10 years in between the two tumor episodes, the methylation is extremely stable. So there are really very few differences. We had some tumors with 10 years in between the primary and the relapse, where we did the methylation analysis, looked for significantly different CPG sites on the two arrays. Couldn't find any single CPG site that was significantly different between the primary and the relapse. So they're not all that stable, but certainly in terms of subtype, the vast majority stay stable between primary relapse. I think that's something you've also seen in medulloblastoma.
Largely.
Seems to be a common feature.
So Lucas Chavez would like to know when the next version of the CNS classifier will be available that will include all of the new subtypes you've described today.
I hope soon. Watch this space. We're expecting within the next maybe 5 to 6 months, I would hope. It's still not quite at the point where we are confident enough. So there are some analytical challenges as well, going from 80 groups up to what is now, including all these different subtypes, we're at around 180 subtypes now of brain tumors in that version, which brings some challenges on the validation side and on the analytical side, but we're working on it and we hope to be able to bring that out soon.
Great. So I think the last question here, have you observed high-grade glioma in the setting of NF1?
Yes, we have. So I think there are two probably major subtypes of NF1 related high-grade glioma in the pediatric setting. One of them is a pretty typical glioblastoma. So those also actually tend to fall into the RTK1 subtype of the pediatric high-grade gliomas, where you really have a very sort of full-blown high-grade tumor. But there's another subset which seems to be enriched specifically for NF1 alterations, and that's something which is now called high-grade glioma or high-grade astrocytoma. It keeps changing the name. So high-grade astrocytoma with pyloid features, I think, is the current consensus, which was previously known as anaplastic pilocytic astrocytoma. So it's with pyloid histology, but clear anaplasia. And that seems to be sort of intermediate between a PA and a true GBM in terms of outcome, but several NF1 tumors in that subset.
Great. So I think we have got through all the questions now, and definitely appreciate you staying on the extra 10 minutes or so. And we'd like to officially close the event permanently now. And thank you again for taking the time out of your evening over there, David.
You're welcome. Thanks again for the invite. And thanks, Paul. Speak to you soon.
Thanks, everyone. Bye-bye.
Bye.